Imagine this, a sudden, just completely devastating outbreak hitting a region, animals getting sick, farms basically shutting down overnight, whole industries just grinding to a stop. And for a while, nobody really knows where it's coming from or how to stop it. This isn't just, a hypothetical. It actually happened quite dramatically in southern England back in 2007. We're talking about the foot and mouth disease outbreaks. They absolutely rocked the region, caused huge disruption, and the economic fallout was, well, hundreds of millions of pounds. But here's the really crucial part and what makes this more than just a story about a disaster. It was a moment where cutting-edge science really stepped up. It transformed what could have been a much longer crisis into essentially a solvable mystery. So our mission today is to sort of unravel the complex story of these outbreaks. We want to focus on how sophisticated scientific tools played this absolute pivotal role in first identifying the source, then tracking the virus's every move, and ultimately informing those critical control measures that got the situation under control. It's like a real-life scientific detective story. And to guide us through all these intricate details, we have technologist Dr. Rama Yusvana, who's a senior lecturer from the Faculty of Engineering Technology at Tun Hussein University of Malaysia, UTHM. His expertise in synthesizing complex information makes a guide for this fascinating deep dive.
Thank you. It's good to be here to talk about this.
Yeah.
It really was a pivotal moment in, veterinary epidemiology. When we talk about foot and mouth disease, FMD, it's not just any animal illness.
Mm-hmm.
It's severe, highly contagious. It's a viral disease affecting cloven-hoofed animals.
Right, so that's cattle, sheep, pigs.
Exactly. Cattle, sheep, goats, pigs, yes, and also their wild relatives.
Mm-hmm.
The virus responsible, FMDV, it's an RNA virus, unenveloped, and it's known for being remarkably robust and fast-spreading. It can just devastate livestock populations incredibly quickly. So in 2007, the UK experienced these two distinct clusters of FMD outbreaks. Both were centered in Surrey, down in southern England. The first confirmed case popped up on August 3rd in a place called Normandy. That really set the alarm bells ringing.
Understandably.
Then, just over a month later, a second cluster started up September 12th, this time in Egham, which is about maybe 20 kilometers away from Normandy.
So close, but distinct clusters initially.
That's right. And the scale of the impact, well, is immense. In total, 1,578 animals were killed on the infected farms. And the FMD virus itself was confirmed in 278 of those cases. Now, this was particularly alarming because, as you mentioned, the UK had gone through that huge FMD outbreak only 6 years before, in 2001.
Yeah, I remember that. The images were just awful.
Exactly. So the memory and the devastating economic cost in that event were still very, very fresh. These 2007 outbreaks caused severe disruption right across the farming sector, and the cost quickly ran into the hundreds of millions of pounds. It was a crisis that needed not just a fast response, but an incredibly precise one.
That's a terrifying picture. And yeah, with 2001 still so raw, the immediate question, the agonizing one for everyone, must have been, where? Where did this come from? Was it, I don't know, a natural thing popping up again? Or were scientists thinking about other possibilities right away? What was the first hint that pointed them somewhere specific?
Well, the crucial breakthrough, and what really pointed investigators in a very specific direction quite early on, was identifying the probable source. It seemed to be a derivative of a specific FMD virus strain, O1BFS 1860.
O1BFS 1860.
Okay, yes. And this particular strain was being handled at two foot-mouth disease laboratories. One was the Institute for Animal Health, the IAH, and the other was Muriel Animal Health Limited. And critically, both of these labs were located on a single high-security site at Pirbright in Surrey.
Pirbright. Wait, how close was that to the first farm?
Just 4.4 kilometers, very close.
Oh, wow. Okay, that's more than a hint then.
It certainly raised immediate suspicion, and the genetic evidence backing this up was incredibly compelling. The virus they found in the outbreak was 99.84% identical in a specific part of its genetic code, the VP1 gene to that FMDV01 BFS 1860 strain from the labs.
Almost identical.
Very, very close. But the real smoking gun, if you like, the definitive fingerprint, was finding a specific genetic marker. A subtle but really significant change at one particular point in the virus's structure, residue VP360. Yes. This specific change, an amino acid substitution from aspartic acid to glycine, it was found in most of the viruses collected from the infected animals in the field. And this particular change provided really strong evidence that a virus adapted to growing in cell culture in a lab was the ancestor of the outbreak virus.
So it wasn't just a naturally occurring virus out in the wild. It had hallmarks of having been grown in a lab.
Precisely. It meant it almost certainly originated from a virus that had been grown and handled in a laboratory setting.
That's not just strong evidence, that feels incredibly precise. So this genetic fingerprinting, it didn't just suggest a source, it basically provided scientific proof. It sounds like it turned this huge mystery into something solvable, with a clear origin. Was there, any pushback on that, or was it pretty much accepted straight away?
Well, scientific consensus always involves careful review and discussion. But the genetic evidence here was so overwhelmingly strong, it really did quickly become the accepted conclusion. It allowed investigators to move past just suspicion to a very, very high degree of certainty. And then, as for how the virus spread once it was out, within each cluster, the spread seemed mostly local. Things like contaminated people moving between farms, maybe equipment, objects, and also aerosols airborne spread over short distances.
Right.
What's interesting is that movements of infected animals themselves were not identified as the main cause of spread between the premises in those local clusters.
Ah, okay, so it wasn't animals being moved around spreading it initially, it was more human activity or local contamination.
That seemed to be the primary driver within the clusters, yes. And then connecting the August and September clusters, there was equally strong genetic evidence. It suggested a probable chain of transmission linking the two, and this chain likely included some undetected intermediate infected premises, places that were infected maybe mildly but hadn't been identified yet, something like what was later designated IP5.
So the virus could have been lurking somewhere unnoticed between the two main flare-ups.
It seems very likely, yes.
Okay, so suspicion is firmly on Pirbright. The virus is spreading. The race is definitely on. How did the scientists, the people on the ground and in the labs, how did they actually piece together the virus's journey? What were the main tools in their toolkit?
It was really a multi-pronged scientific approach. Yeah. Think of it like different teams in a detective agency working together. First, you had the clinical investigations. Absolutely vital. This meant vets going onto the farms, doing detailed animal inspections, looking for those classic signs of FMD, the blisters, the lesions. And crucially, they tried to estimate the age of those lesions.
Why the age? What does that tell you?
It gives you critical clues about the timeline. When did the infection likely arrive on this farm? It helps trace the spread back in time.
Right, okay. Building the timeline.
Exactly. And at the same time, you had rigorous laboratory investigations running in parallel. Samples were being collected from affected animals, bits of tissue, blood, throat swabs, that kind of thing. These samples were then tested using various methods to confirm the virus was there. They looked for the virus protein, the antigen, using immunological tests. And really powerfully, they detected its genetic material using real-time RT-PCR.
RT-PCR, that sounds familiar. Is that like the COVID tests?
Very similar principle, yes. Think of RT-PCR as a super-sensitive test. It can find tiny, tiny traces of the virus's genetic blueprint, its RNA. And the key thing is it can often detect the virus even in animals that are infected but aren't showing any symptoms yet. Preclinically viremic animals.
The silent spreaders.
Precisely. Catching them early is absolutely crucial for control. And finally, they also did blood tests to look for antibodies, which show if an animal has mounted an immune response to the virus.
Okay, so clinical signs, virus detection, antibody tests, a whole suite of lab work.
Yes, but what's truly fascinating here, and what was the real game changer in this investigation, was the power of genetic analysis, what we call molecular epidemiology. Initially, scientists looked at just one small but characteristic part of the virus's genetic code, that VP1 gene we mentioned earlier.
The quick ID check.
Exactly. It told the general type of virus, confirmed it was O1BFS 1860 related. But it didn't have enough detail, enough resolution to track its exact movements within the outbreak. It couldn't distinguish subtle differences between viruses from different farms easily.
Right, so it tells you it's the right family, but not the specific individual.
Sort of, yes. The real breakthrough was full genome sequencing. This is where they sequenced the entire viral genome, all 8,300 or so bases of its RNA. And critically, they did this in real time as the outbreak was unfolding. It was like moving from, blurry black and white photos to having a live high-definition video stream of the virus's journey.
Wow.
This gave us just unprecedented high-resolution data on how the virus was evolving, tiny changes happening as it spread, and mapping its exact transmission pathways.
That's an incredible leap in detail. So instead of just knowing the general type, you could see its specific family tree almost, and how it was subtly changing as it moved from farm to farm.
Exactly that. You could build these transmission trees.
So how did that level of detail help confirm the lab origin theory beyond just that initial VP360 marker you talked about?
Well, having the full genome allowed us to look at other specific amino acid residues, other points in the genetic code. For instance, we could analyze changes at sites like VP2134 and another one, VP356, alongside that key V360 site. By looking at the patterns of changes across these sites, we could get a clearer picture. We could see if the virus showed signs of adapting to cell culture in the lab.
Right.
And then, importantly, if it started to revert back towards a more wild-type configuration once it got back into replicating in actual animals.
Ah, so it changes in the lab and then changes back slightly when it's in an animal.
That's the idea. The VP360 change to glycine was that strong initial indicator of cell culture adaptation. But then, observing reversions, say at VT356, where it might flip back to the form usually seen in animals, that provided fairly compelling evidence. It was consistent with the virus originating in a lab culture and then spreading through the animal population.
So it's like tracking its adaptation history.
Exactly. This detailed genetic fingerprinting really helped solidify the lab origin theory and allowed scientists to track these nuanced changes as the virus moved farm to farm. If we connect this to the bigger picture, this real-time genetic analysis was absolutely critical. It directly influenced the control measures being implemented. First, and maybe most importantly, it decisively confirmed the Purebright laboratories as the most likely source. That was just essential information for the public health and animal health authorities to have and to act upon.
Gave them certainty, focus.
Exactly.
Yeah.
But beyond just identifying the source, this data allowed for incredibly precise tracking, farm-to-farm tracking of the virus's movement. It let investigators identify the probable chains of transmission events. Who likely infected whom?
Building that transmission tree in real time.
Yes. But it wasn't just about confirming what had already happened. This is where it gets really interesting. It was also about predicting what might happen next. The genetic data, for instance, actually predicted the existence of those undetected intermediate infected premises we talked about earlier.
It predicted them. How?
By looking at the genetic links. If virus from farm A was closely related to virus from farm C, but slightly different, the genetics might suggest there had to be an intermediate step, a farm B, even if farm B hadn't been found yet. And what's truly remarkable is that subsequent field investigations, boots on the ground, then found those predicted locations, like IP5.
That's incredible. So the lab work was telling the field teams where to look next.
In a way, yes. It's a really powerful demonstration of predictive science in action during an outbreak, moving from just reacting to actually having some informed foresight.
That predictive capability sounds truly groundbreaking. It's almost like the virus's own genes were giving you a real-time map of where it was likely headed.
That's a good way to put it. And these insights also profoundly informed the ongoing field investigations. It helped them figure out if new cases represented brand new sparks from the source or if they were just part of the ongoing spread.
Ah, okay. Single source versus ongoing transmission.
Exactly. For example, the genetic differences they saw between the August and September clusters showed pretty clearly that the second cluster was a result of ongoing transmission spilling over from the first. It wasn't a second, completely independent leak from Purebright.
Which must have been crucial for deciding how to manage the response, right?
Absolutely vital. Knowing it was one continuous chain, albeit with hidden links, helps direct resources and understand the true dynamics of the outbreak. So ultimately, all this rapid, precise genetic information, looking at the virus's movement and its evolution in near real time, directly influenced and shaped the control measures. It genuinely transformed disease management from being purely retrospective, looking back after the event, to enabling real-time intervention during the crisis itself.
The 2007 FMD outbreaks in southern England, they were such a stark reminder, weren't they, of the ever-present threat of animal diseases, how quickly things can escalate, the economic devastation, the disruption. But as we've really unpacked today, it was also this profound testament to how advanced genomic science has just revolutionized our ability to understand, to track, and ultimately to combat these pathogens in real time. The ability to pinpoint the source with such certainty, to follow those invisible pathways of transmission, even to predict where new infections might pop up based purely on the virus's genetic code. It just fundamentally changed our response capabilities. It really makes you wonder, doesn't it? Thinking about the lessons from 2007, how prepared are we now for future outbreaks? Not just of FMD, but really any rapidly evolving pathogen, whether it affects animals or humans? And what crucial role will this kind of real-time molecular epidemiology continue to play in safeguarding, well, global health and our economies? It's a big question. We owe a huge debt of gratitude for the enlightenment and invaluable insights shared today by technologist Dr. Rama Yusfana, senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia, UTHM. Your insights have truly enlightened us. Thank you so much for walking us through that.
It was my pleasure. A fascinating, though concerning case study.
Absolutely. We hope you, the listener, will continue your own deep dives into fascinating topics. Thanks for joining us, and we'll catch you next time.